Hello, today we are going to study Batrachospermum part 2, in which we are going to study reproduction in Batrachospermum and life cycle pattern as well as systematic position. So the first point is reproduction in Batrachospermum. The gametophyte of Batrachospermum reproduces only by sexual method, whereas asexual reproduction is confined only to Cantrantia stage, which is a new stage in the life cycle of this red alga. So first, sexual reproduction. This red alga Batrachospermum reproduces sexually by means of highly advanced oogamous type of reproduction, and the plant of Batrachospermum may be monoecious, that is homothelic, in which both male and female sex organs they are present on the same thallus, or in some cases you can have a dioecious condition or heterothelic condition in which you can get separate male and separate female plant. The male sex organ as well as female sex organs are well developed. The male sex organ is called as spermatangium, which is nothing but it is a basically antheridium, and the female sex organ is a carpogonium, comparable with the oogonium of other members of algae. Both the sex organs they are produced at node and either on ordinary or specialized short branches that are always produced at the node of main axis. So we have to study the structure of both male as well as female sex organ. First, male sex organ, that is a spermatangia, plural, singular spermatangium, which is also called as antheridia or antheridium. The male sex organ is a antheridium or spermatangium, and these antheridia or spermatangia are usually born in clusters on short spermatangial branches. If you observe here, this is a specialized branch which is called as either antheridial filament or spermatangial branch, and it is always produced at the node where you can have a glomerule. So some of the branches of glomerule or some specialized antheridial filaments they are produced at the node of the main axis of Batrachospermum, and usually these antheridial filaments or spermatangial branches produces antheridia or spermatangium. The production of antheridia or spermatangia is either in this singular manner, or they may be produced in groups or clusters, and always at the tip of the cell from which they are produced. When it is produced on apical cell, they are apical in position. If they are produced at the intercalary cell, they are usually lateral in position. So the position of spermatangia is a variable. If you observe the structure of spermatangium or antheridium, it is a more or less spherical or elongate, spherical or globular structure, and the content of this spherical or globular spermatangium get developed into a single non-motile, non-flagellated male gamete that is called as a spermaceum. This is a spermaceum. So when this antheridium get mature, that is at the maturity of spermaceum, a slit to the wall of spermatangium is developed. That slit grows and the mature spermacea they are directly released in water, and these spermacea, which are unicellular, uninucleate, non-flagellated and non-motile, one usually swims in water along with the water current. That is the passive movement and not active movement. And due to this passive movement, these spermatia, they are usually brought to the female sex organ, that is carpogonium. So the second sex organ, that is carpogonia, which is a female reproductive organ, and this female reproductive organ is called as a carpogonium in case of Batrachospermum. Usually these carpogonia are unicellular. If you observe this structure, these are the developmental stages, and this is the mature carpogonium. This carpogonium is a unicellular structure, and it is a large flask-shaped structure. This is the basal swollen portion, and this is a long, narrow, elongated neck. So each carpogonium has two distinct portions, the basal swollen portion and a narrow, elongated neck, which is called as trichogyne, and this basal swollen portion is called as a oogonium proper. Now here this basal swollen portion is a oogonium proper, and it develops into a single non-motile, non-flagellated egg, which is a female gamete. Whereas the trichogyne, which is a large tongue-shaped or triangular structure, the shape of this trichogyne varies from species to species, but this trichogyne is a enucleate. That means it lacks nucleus. Cytoplasm is present there, but nucleus is absent in this trichogyne. As this basal portion, that is oogonium, produces the egg, it is a fertile, whereas this is a enucleate structure or nucleus is absent, so it is a non-fertile, or you can say it as a sterile structure. And the main function of this sterile trichogyne is to receive the male gamete, that is a spermatia, for the act of at the maturity. So next point is a fertilization. As I have told you that the mature spermatia swimming in water get attached itself to the sticky trichogyne of mature carpogonium. That means this carpogonium towards maturity produces a sticky substance along the wall of trichogyne. So these are male gametes, that is spermatia, get easily attached to the wall of trichogyne. When such spermatia get attached to the trichogyne, the contact wall between the spermatium and trichogyne get dissolved, and the male nucleus migrates into the trichogyne. And this male nucleus ultimately travels towards the female gamete, that is egg nucleus, in a downward direction through the cytoplasm of trichogyne, and ultimately it is brought to the surface of egg nucleus, which lies side by side. Once this male nucleus comes in a contact with the egg nucleus or female nucleus, they get paired, and after the pairing of male nucleus and female nucleus, here a mucilage plug is developed. Now this formation of mucilage plug is to avoid the entry of additional male nucleus or male gamete for the fertilization of this female nucleus or egg nucleus. So mucilage plug formation is important once the pairing of one of the male nucleus with the egg nucleus. So here this pairing of male nucleus with the female nucleus immediately get result in the union or fusion of both the two nuclei. This union or fusion of male nucleus with the female nucleus is called as fertilization, and the product of fertilization is a formation of diploid zygote. So such a diploid zygote is produced due to the process of fertilization. Now this diploid zygote, once get formed, usually undergo the process of germination. So germination of zygote or formation of cystocarp is the next stage in the life cycle of Batrachosperma. Now this diploid zygote that is produced within the carpogonium at base immediately undergo first reduction division or meiosis. So due to this meiosis, the diploid zygote nucleus results into formation of 4 haploid nuclei. So zygote nucleus, which is a diploid, upon reduction division produces 4 haploid nuclei, and at the same time these 4 nuclei, they are usually produced here in the basal fertilized carpogonium. Immediately 4 protuberances or outgrowths they are produced on the wall of this fertilized carpogonium, and one such nucleus enters into each protuberance or outgrowth. So one daughter nucleus migrates into each protuberance, and therefore once this nucleus get entered into the carpogonium, usually septa formation takes place and the protuberances or outgrowths, they are cut off from the wall of the carpogonium or fertilized carpogonium. So such a single celled structure that is developed on the wall of carpogonium is called as gonimoblast initial or gonimoblast initial cell. Now this gonimoblast initial develops, and they are 4 in number. Each such initial develops into a short filament due to repeated mitotic division, and such a short filament is called as gonimoblast filament, and the apical cell of this gonimoblast filament is producing carposporangia at its tip. Now each such carposporangium is unicellular, and again it produces haploid unicellular spore called as carpospore. So here, when this development of zygote takes place into the gonimoblast initial cell, gonimoblast filament and carposporangia producing carpospore takes place, hypobasal cell of carpogonium produces many sterile filaments, and all these sterile filaments usually covers the gonimoblast filament, carposporangia, carpospores, and it results in the formation of a compact structure which is called as cystocarp or carposporophyte. So each such carposporophyte is a haploid stage in the life cycle of Batrachospermum, but this haploid stage is attached to the gametophyte of Batrachospermum, so it is dependent on gametophyte for its nutrition. So it is not independent phase in the life cycle of Batrachospermum, but is dependent on the gametophyte of Batrachospermum. Now each such carpospore produced within the carposporangia towards maturity get liberates by formation of slit to the wall of carposporangium, and such liberated carpospore, when it liberates in water, along with water current they are carried to distant places, and when it falls on a suitable substratum, it germinates to produce a new haploid stage in life cycle of Batrachospermum, and that stage is called as a Cantransia stage, which is a very important stage in the life cycle of Batrachospermum. Now here, these are the carpospores, and these carpospores, when falls on a suitable substratum during favorable condition, germinate to produce such a small heterotrichous filamentous structure. Now this heterotrichous filamentous structure apparently looks like another alga. The name of that alga is Cantransia. So this phase in the life cycle of Batrachospermum apparently looks like a Cantransia alga, and therefore this phase is called as Cantransia phase. So Cantransia phase is the haploid phase in the life cycle of Batrachospermum, in which you can have a heterotrichous filamentous structure. Some of the filaments, they are prostately growing, forming prostrate system, and some of the filaments, they are erectly growing so as to form erect system. And such heterotrichous filamentous alga is also there. The name of that alga is Cantrantia. As this phase in the life cycle, or this heterotrichous filamentous structure apparently looks like Cantrantia, so this stage is labeled as Cantrantia stage. Now this is again a haploid phase in the life cycle of Batrachospermum. Now this Cantrantia stage reproduces asexually by producing monospores within the monosporangium. What happens? These erect system or filaments of the erect system produces lateral branches, and at the tip of such lateral branch, either one or such two spherical specialized structures are produced, which are called as a monosporangia. The name itself indicates that each structure is producing a single spore. Mono means one. Sporangium, that is producing spore. So each such sporangium as produces a single spore, it is called as monosporangium, singular, plural monosporangium. Now the content of this monosporangium develops into a monospore, which is again a haploid uninucleate structure, and towards the maturity of the monospore, the wall of monosporangium again splits and the monospores, they are directly released in water. But the germination of monospore again results into formation of new Cantrantia stage and never Batrachospermum in any case. So here, due to the asexual reproduction by monospore, this Cantrantia stage get multiply in number, and monospore in any case doesn't produce the adult gametophyte of Batrachospermum. So each such monospore again produces an individual Cantrantia stage. Then you will ask me, what about the phase of or fate of this Cantrantia stage in the life cycle of Batrachospermum? Usually towards the maturity, here adult shoot or adult gametophyte of Batrachospermum usually develops from the lateral sides of the erect system of this Cantrantia stage. So each this structure that is developed from the cells of the erect system is a adult gametophytic thallus of Batrachospermum. So this is a very important phase or stage in the life cycle of Batrachospermum, which is called as a Cantrantia stage, and this Cantrantia stage is an additional phase in the life cycle of Batrachospermum, which is not present in other red algal members. So this is about the Batrachospermum life cycle in which Cantrantia stage is present, which we have observed in a earlier slide diagrammatically. So here, from the mature Cantrantia stage, adult Batrachospermum develops from the erect system of Cantrantia. Then the next point is a life cycle of Batrachospermum. If you observe this life cycle of Batrachospermum, here this is the adult gametophyte. This adult gametophyte reproduces sexually by means of highly advanced oogamous type of sexual reproduction that results into formation of zygote. Then this zygote undergo reduction division, and here the haploid generation, new generation in the life cycle is formed, that is called as carposporophyte or cystocarp. That carposporophyte produces carpospores. These carpospores germinate to produce a new phase in the life cycle, that is Cantrantia stage. This Cantrantia stage reproduces asexually by monospore and again produces Cantrantia stage, and towards the maturity, adult gametophyte thallus of Batrachospermum is developed from the erect system of this Cantrantia stage. So if you observe this graphic life cycle, this adult gametophyte, which is a haploid structure, is one phase in life cycle. Then second phase is a haploid carposporophyte, which is totally dependent on gametophyte. Here it has been labeled as a cystocarp or carposporophyte. They are the synonyms. Then third phase in the life cycle is again haploid Cantrantia. So these three haploid phases, they are regularly alternating with only a short-lived diploid structure, that is zygote. So this type of life cycle is called as triphagic. Why it is called as triphagic? Because there are three different phases. One, that is gametophytic thallus. Second one is a carposporophyte or cystocarp, and third one is the Cantrantia stage. So it is called as a triphagic. And this triphagic type of life cycle is of haplontic type. One, haplontic is for gametophyte. Second, haplo is for carposporophyte or cystocarp, and third for this Cantrantia stage, and therefore it is called as haplo. This is a gametophytic. Haplo, this is the carposporophyte or cystocarp, and haplontic, here it is a third phase in the life cycle, that is a Cantrantia stage. Diploid stage is a highly reduced, and it is restricted only to a short-lived zygote. Then last point is a systematic position. This is belonging to group algae because they are autotrophic thalloid plants. Division Rhodophyta, red algae, due to characteristic chlorophyll A, D, and predominance of R-phycocyanin and R-phycoerythrin. Then class Rhodophyceae, in which you can have all plants, they are without motile stages in a life cycle. Order Nymallionales, family Batrachospermaceae, and genus Batrachospermum. Thank you.